Okay, so hello, I'm Philip, and I'm an undergraduate placement year student, and today I'll be talking about what I've been doing, and this has basically been producing lots of PGK for NMR experiments. So when I mention PGK, I'm really talking about the specific isoform, human phosphoglycerate kinase 1. And as a quick reminder, PGK is a very important enzyme in glycolysis and gluconeogenesis, responsible for the interconversion of 1,3-bisphosphoglycerate and ADP to glycerate 3-phosphate and ATP. So to boil this down even further, it's simply a transfer of a phosphoryl group between the substrates. So this is the structure of PGK. It's composed of 416 amino acids, which constitutes around 45 kilodaltons in molecular weight. It can then be roughly divided into two lobe domains. These aren't the exact divisions, but are close enough. These domains are the C-terminal and N-terminal domains, respectively, and are primarily linked together by an alpha helix, namely helix 7. And on the N-terminal domain, we have a substrate binding site for the glycerate 3-phosphate or the 1,2-bisphosphoglycerate. And then mirroring this, we have the C-terminal domain, which has a nucleotide substrate binding site for ADP or ATP. And this binding is mediated by a magnesium 2+ ion, which helps stick the nucleotide to the enzyme. And in order for PGK to catalytically function, it's been proposed that a hinge movement at helix 7 results in the enzyme changing to a closed conformation, moving the two binding sites closer together, allowing for a successful reaction. So why am I purifying all this PGK? Well, the main reason for doing it is for Emma's master's project, which is looking at the maturation of the drug remdesivir. Remdesivir is a drug that's used to treat viral infections by causing premature dissociation of viral RNA from RNA-dependent RNA polymerases. They can only do this in its triphosphate form, shown here as NUC-TP. But this form has a hard time getting into cells due to its many charged phosphate groups. As a result, remdesivir is administered in its nucleoside form or its monophosphate form, and this monophosphate form is masked using proteid technology. So therefore, figure 1 here shows the overall proposed pathway that remdesivir takes to mature into its active triphosphate form. And in the final process, from diphosphate to triphosphate, PGK is thought to catalyze this step. As a side note, remdesivir is very similar in structure to ATP, so it makes sense why PGK has been proposed for this role in its maturation. So the purification method that I used was that from ex-PhD student Jalgis's thesis. The protocol can be boiled down to this flowchart, and as you can see, there are two main purification steps, those being ammonium sulfate precipitation and cation exchange chromatography using the ACTA purification system. And it's these two steps that I will be discussing in further detail. So despite ammonium sulfate precipitation being an older technique, the primary reason for using this is because it has been shown to work pretty well with PGK. Apart from that reason, some other reasons are that AS is very cheap, very soluble, and stabilizes protein structure quite well. And this AS precipitation was carried out in two stages. The salting in stage saw the sample become saturated with 40% AS. The insoluble content was then centrifuged out. And the salting out stage bumped the AS saturation to 80%, resulting in the precipitation of proteins, including that of PGK. And here is the gel taken from the AS precipitation. The second row shows the supernatant retrieved from sonication of the cell extract. The third row shows the pellet from that same sonication, and the fifth and sixth rows correspond to 40% AS and 80% AS, but due to ammonium sulfate's different conductivity, the 40% line was wobbly and the 80% didn't show up at all. This was fine, however, though, as you can see in lane 6, after the AS purification step and subsequent dialysis of the remaining AS, we have a nice cleaned up sample compared to lane 2. And this was ready for the ACTA purification. So the final major step in the purification process was doing cation exchange chromatography with the ACTA. The specific column used was a CM Sephiroth cation exchange column. Just some details here. We've got PGK's isoelectric point, and that was about 7.28. So to accommodate this, the buffers used were pH 6.2 to give PGK its overall net positive charge. PGK was then eluted using a 1 molar sodium chloride salt gradient, and as you can see here in figure 3, you can see a nice spike showing where PGK had been eluted. And finally, here is the gel representing the fractions containing PGK. Lanes 2 and 3 are just the flow-throughs from start and end of the process, whilst lanes 5 to 11, especially lanes 6 and 7, show quite a bit of PGK being extracted. So just as a summary, the main reason I've been purifying PGK is for the remdesivir project. PGK is thought to catalyze the last 4th step in the maturation of remdesivir, and ammonium sulfate precipitation and actor cation exchange chromatography are effective methods for purifying PGK. As for more work in the near future, we're planning to run a TROSY experiment to check the purified PGK sample, and we'll hopefully be forming some PGK crystals to try and cross-soak with remdesivir. And finally, of course, I'll be producing even more PGK, and that is for a different project.